Okay, come back to the NCERT. Then we start the another process. Now, second point is our transcription. Transcription. How a transcription means what? Formation of messenger RNA, right? That is called transcription. With the help of DNA, we can say in the process of transcription, formation of messenger RNA with the help of DNA. Means DNA codes the DNA give the code to messenger RNA, right? That is transcription. The process of copying genetic information from one strand of DNA into from one strand of DNA. Here, this transcription it occurs only in one strand of DNA, not in two, right? One strand means that is called template strand, and that is 3'5' strand, right? Only one, right? The process of copying genetic information from one strand of DNA into RNA is termed as transcription, right? Copying genetic information from one strand of DNA, right, into RNA, into mRNA or RNA, it is called transcription. Here also, the principle of complementarity governs the process of transcription. That complementarity is here also, right? This transcription is totally depend upon the complementarity. Complementarity means according to the nitrogen base. Except the adenosine, one exception is there, because adenosine it binds with thymidine in case of DNA replication. But here what happens? In RNA, thymine is absent, so here it binds with uridine triphosphate. Except of adenosine complements, right, now forms base pair with uracil instead of thymine. We know very well, right? Because thymine is not present in RNA, so here uracil binds with. That means what? Here the 4 nucleoside triphosphate which are used in this case, they are not same as to DNA, because one is different. In DNA we know deoxy nucleosides are there, deoxy nucleotides we use there, but here what we use? Here we use A, MP, G, MP, C, MP. These 3 are same, but in place of TMP, so TMP is not present here, TMP it is not present here, but in place of TMP it is UMP, that is uridine monophosphate, right? So these are monophosphate which are used in the formation of RNA, right? But they are available in the form of, they are also available in nucleoplast, but it is not in the form of monophosphate, but they are present in the form of triphosphates. So triphosphate is what? Here ATP, GTP, CTP and UTP. Adenosine triphosphate, guanosine triphosphate, cytidine triphosphate and uridine triphosphate. They are available in the triphosphate. Same process here also. They act as dual purpose. Dual purpose means they act as substrate, and another function is they provide energy for the formation of or for the transcription, for the formation of messenger RNA or for transcription. Okay. Now see this one. Unlike the process of replication, which one set in the total DNA of an organisms get duplicated, in transcription. Transcription means we know very well euchromatin part of DNA is responsible for transcription because it is transcriptionally active. Heterochromatin it is transcriptionally inactive. That means what? The whole DNA is not here. Whole DNA is not responsible for the formation of messenger RNA, or we can say whole DNA never forms transcription, right? Only a specific part is responsible for transcription. Okay, and this part we understand, this one is euchromatin part of DNA. Only a segment of DNA and only one strand is copied into RNA. One strand means which strand? It is 3'5' strand. Suppose these 2 strands are there DNA, right? These are hydrogen bonds. right? So this one is 3 dash, 5 dash, and this one is 5 dash, 3 dash. So this strand 3 dash, 5 dash, this one is called template strand, and it is responsible for transcription. 3 dash, 5 dash. This strand is called template strand, right? Now here, this necessitates defining the boundaries that would demarcated the region and the strand of DNA that would be transcribed. Why only, now question is coming, why only one strand is responsible for transcription? Why not both? Right? Because when these both starts transcription, then what happens? A different type of messenger RNA are formed, right? Because now sequence is totally just opposite, and totally opposite means a different protein is formed. Okay, it is given here. We can read here this. Now question, why both the strands are not copied during transcription, has the simple answer. First, two things are given here. One is, first, if both strands act as template, they would code for RNA molecule with different sequence. Right? Because both strands, they have different amino acids, different base. Okay? You can understand, suppose that a strand is AUG strand is there, that means what? In another strand, what is there? It is not AUG, but it is UAC. So both are different nitrogen base, so they form different strand. One RNA strand is from UAC, another strand is from AUG, and UAC and that is UAC, both are responsible for different amino acids also. They are genetic codes and they are responsible for different amino acids. That means what? A different protein is formed. From a same, means we can say same site of DNA, two different type of RNA strand are formed. First thing is here, because remember, complementarity does not mean identical. It is totally different, right? And in turn, if they code for protein, the sequence of amino acid in proteins would be different. As I will tell you, AUG means methionine, right? UAC is not a methionine, right? UAC means what? It is valine. UAC means it is responsible for valine, right? So it is not responsible. Hence, one segment of DNA would be coding for two different proteins. A single same segment. Two different proteins are formed, and this would complicate the genetic information transfer machinery. Right? Two different type of proteins are formed at the same time, and these two different proteins they have different functions. Right? It becomes more complication. Second, the two RNA molecules, if produced simultaneously, then what happens? They would be complementary to each other, and when they are complement to each other, they form hydrogen bond in between them. Now they becomes double stranded, and when they become double stranded, they becomes inactivated. Right? That is given here. Because they are complement to each other, hence would form a double stranded RNA, and this would prevent RNA from being translated, because it is double stranded RNA, right, into the proteins, and the exercise of transcription would become a futile one. Do you understand here? Two regions are given here. First region, if both strands they form, both strand is transcribed, both strand forms messenger RNA, so their nitrogen base means we can say sequence of nitrogen base is totally different. Because their sequence of nitrogen base is different, that means genetic code is different. Genetic code different means what? Amino acids. Sequence of amino acid different means different proteins are formed, and which create a complication in transcription, because two different type of proteins are from a single segment of DNA. Right? Second, what is given here? If two strands are formed, so that means they are complement to each other. That is right, because DNA is complement to each other. They are complement to each other means what? They forms hydrogen bond, and now this DNA becomes double stranded. Double stranded means now it is like DNA, it is not active, right? Because DNA is not active, double strand DNA is not active. When RNA becomes double active, it is also not active. It becomes a stable structure. That's why only single strand is formed. right? And these two regions are given here. Now, transcription unit. Another point is our transcription unit. A transcription unit in DNA is defined primarily by the three regions in the DNA. Which three regions are there? Promoter. First region is our promoter, right? Structural gene and terminator. Three regions. Promoter, structural gene and a terminator. Promoter is responsible for the binding of RNA polymerase. RNA polymerase enzyme, it binds with promoter. Structural gene, it is responsible for elongation, right? Third, elongation of protein, elongation of RNA molecule, right? And third, terminator means is responsible for the stopping the process, to stop or terminate the process. It is responsible to terminate the process. Sometimes what happens in promoter and in terminator, there is some factors. As in case of prokaryotic cell, we can say sigma factor is present at promoter site and the terminator aspect is rho factor is there. Okay, now coming to the next page. Now you understand a transcription unit, it has three regions. Which regions are there? It is promoter, structural gene and terminator. Promoter for initiation, structural gene for elongation and terminator means for terminate the process or for stop the process. Now, there is convention in defining the two strands of the DNA in the structural gene of a transcription unit, since the two strands, they have opposite polarity and the DNA dependent RNA polymerase, because here RNA is formed, RNA polymer is formed, so this enzyme is called as RNA polymerase. It is totally DNA dependent because code comes from DNA, so it is called as DNA dependent RNA polymerase. When in DNA replication, what is there? It is DNA dependent DNA polymerase. One thing also there, suppose that someone is asked, in case of retrovirus, in previous lecture I tell you, retro means what? Reverse transcriptase. That time which type of enzyme is there? It is also DNA dependent DNA polymerase? No, because in retrovirus what happens? RNA forms DNA. Because RNA forms DNA, this enzyme is called RNA dependent DNA polymerase. Because DNA polymerase is formed, it is called DNA polymerase, and it is RNA dependent, so it is called RNA dependent DNA polymerase in case of retroviruses when RNA forms a DNA. When DNA forms DNA means DNA dependent DNA polymerase. When DNA forms RNA, it is called DNA dependent RNA polymerase enzyme. Okay. Here, since the two strands have opposite polarity and the DNA dependent RNA polymerase also catalyze the polymerization only in one direction, and that direction is 5'3', right? The strand here, one direction, 5'3" direction, right? Two strands, they have opposite polarity, first thing is there, and DNA dependent RNA polymerase also catalyze the polymerization in only one direction, that is 5'3". The strand that has the polarity of 3'5", here you see 3'5" act as a template strand, is also called as sense strand, and it is also referred to as template strand because new messenger RNA is formed on this strand, right? Second, the other strand which has the polarity of 5'3", same as to this polymerase enzyme, and the sequence are same as the RNA, but one difference is there because in RNA uracil is present in place of thymine, right? Except thymine at the place of uracil, right? Because in RNA uracil is there, thymine means it is present in DNA, right? Now it is displaced during the transcription, right? Strangely, because these codes are matched with RNA code, so it is called as coding strand, but is given strangely this strand which does not code for anything is referred to as coding strand, right? Coding is same as to RNA because it contains same code as RNA, only one exception is there, it contains this DNA strand, it contains thymine in place of uracil, only this change, right? Remaining all the codes, they are same sequence. So it is called as coding strand. All the references point to while defining a transcription unit is made with coding strand, right? To explain the point hypothetical sequence from a transcription unit is represented below. Here a means this basic sequence is given, nitrogen sequence is given, that one is template strand, and then that one is coding strand. Now here question is given, can you now write the sequence of RNA transcribed from the above DNA? Your answer is yes, easily. How easily? Because we know this 5' 3', it is the coding strand, it has the same sequence which is present in the new transcript RNA, but one thing is there, we have to, this in place of T it is U, in place of T it is U, in place of T it is U, in place of T U, U and U. Means we can say UACG, 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 that is the RNA. This one is the transcript RNA. Do you understand? Very easy. Only we have to see the 5' 3'. Okay, suppose that only one this template strand is given. So we need the this process of complementarity. That A means is this T, because T is absent in RNA. So we should write here U. T means A, G means C, C means G, A means T, T means T is absent in RNA. So we use here U. Okay. Do you understand? Now a figure is given. That is a transcription unit. It is the figure of transcription unit. Here in this figure, you can understand the means A. We can say here this is the DNA, right? This one is DNA. You can see here this one is promoter site. Promoter in this one is first. This one, this strand is called. This one is. It is called coding strand. And this one is our, sorry, this one is not a coding, this one is called as template strand. Template strand, and this one is coding strand. This one is coding strand, because this 5' 3' and this one is 3' 5'. And this promoter when we consider the template strand, the promoter is toward the 3' strand. But when we consider the coding strand, that time what happens? Promoter is located toward the 5' strand. This side is terminator. Terminator means coding strand. It is toward the 5' strand. But coding strand it is toward the 3' strand. But in case of template strand, it is the 5' strand. Okay. The promoter, structural gene, and terminator. Right here, the process starts from this to that. This is the direction of the process. Formation of messenger RNA. Right? Means messenger RNA from this side to this. Okay. The promoter and terminator flank the structural gene in a transcription unit. The promoter is said to be located toward the, that is, promoter is said to be located toward the 5' end. When this look 5' end, 5' end means what we describe when we consider this coding strand. Then 5' end, that is called upstream of the structural gene. The reference is made with respect to the polarity of coding strand. With respect to the polarity of coding strand, that's why it is said here it is toward the 5' end. And when we respect the template strand, that is toward the 3' strand. Right? It is a DNA sequence that provides binding site for RNA polymerase. Binding site for RNA polymerase. Right? In prokaryotic cell, this site is called Pribno box, but in case of eukaryotics, they are called as Tata box, or we can say this one is Hogness box. So this promoter site, it is made up of 6 sequence of nucleotides in case of prokaryotics, but in case of eukaryotics, it is made up of 7 nitrogen base. Okay, and it is the presence of promoter in a transcription unit that also defines the template and the coding strand. Okay, who is responsible? That promoter site. Promoter site, it defines the which one is coding stand and which one is template stand, right? Promoter is present. The site where promoter is present, when we consider the template stand, it is 3 dash. But when we consider that is coding stand, it is 5 dash. So promoter is responsible. The presence of promoter is responsible to describe the which one is template and which one is coding stand, right? By switching its position with terminator, the definition, that is by switching of its position with terminator, what happens? The definition of coding and template stand should be reversed. The terminator is located toward the 3 dash end of coding stand, that is downstream of the coding stand, and is usually defines the end of the process of transcription. That's why it is called as downstream process. And because transcription is initiate at the 5 dash end, so it is called as upstream, right? There are additional regulatory sequences are also there that may be present further upstream or downstream. They are present before the initiation codon, or we can say before the initiator, after the terminator, right? Downstream to the promoter. Some of the properties of these sequences shall be discussed while dealing with the regulation of gene expression. When we read the lag operand model, at that time we will understand what is the function of these. We can say it is non-coding stands, right? Which one is just before present before the promoter and after the terminator. So they affect the transcription. Okay. So this one is our transcription unit. Transcription unit means we have 3 parts. One is promoter, right? Another one is structural gene, and last one is terminator. Promoter means it is responsible for initiation. Second one is our structural gene, which is responsible for the elongation of chain. Third one is our terminator, for stopping or for terminate the process, right? Another thing is given here. When we consider the promoter is located toward the 5 dash end of coding stand, or we can say 3 dash end of non-coding stand, that is template stand. That is terminator, it is located toward the 3 dash end stand of coding, right, stand. And this terminator, it is located on coding stand toward the 5 dash end, right? Okay. Now transcription unit and the gene. What is given here in transcription unit? A gene, our very previous, when I was learning in the that means we can say intermediate classes, at that time I learned that a gene is a small fragment of DNA in which at least one genetic trait is present at that time, but this time it is not correct, right? It is here given a gene is defined as the functional unit of inheritance. At that time also I read gene is the functional unit of inheritance, right? But definition is now changed. Though there is no ambiguity that the gene are located on DNA, it is difficult to literally define a gene in terms of DNA sequence. That's why I tell you what I read at that time, but today it is totally different. At that time I read in humans at 1,20,000 genes are present at that time, because at that time genome project is going on in 1994, 1992, not 94, in 1992. And we know genome project, it starts from 1990 and it is completed in 2003, right, on paper, but actually it is completed in 2006 May, right? So before that means at that time human genome project is not completed, and at that time we understand that human genome, it contains at least 1,20,000 to 1,50,000 genes are there. So I read at that time 1,20,000 genes are present in human, but really we have only 30,000 genes are there. At that time I read gene means what? It is the smallest fragment of DNA in which at least one genetic trait is present, but this time it is wrong. So in new NCERT or in no new definition means a gene represents in which three things are there, that is cistron, mutant and recon, right? And most clearly it is given here. The gene, this time gene cannot be defined with DNA. Here it is given. It is difficult. What is given? It is difficult to literally define a gene in terms of DNA sequence. Why? The DNA sequence coding for transfer RNA or ribosomal RNA molecules also define a gene, right? However, by defining a cistron means what? A functional unit is called cistron, right? As a segment of DNA coding for polypeptides, this is a segment of DNA which is coding for a polypeptide. The structural gene in a transcription unit could be said as monocistronic, and monocistronic mostly present in eukaryotic cells. Monocistron means only one functional unit is there. Polycistron means, poly means many and cistron means functions, right? That is in mostly in eukaryotes, and or polycystonic, which is mostly present in bacteria or prokaryotes, right? That is prokaryote, mostly in eukaryotic means monocystonic, and polycystonic mostly in bacteria or prokaryotes. So when we use cistron for defining the DNA, segment of DNA, right? So one is monocystonic, another one is polycystonic. In eukaryotes, the monocystonic structure genes have interrupted coding sequence. Intron exon is there. The gene in eukaryotes are split genes. Because why it is called interrupted? Because we know they are alternatively arranged in the form of intron, exon, intron, exon, intron. Intron means it is non-functional unit, exon means functional unit. Then after what happens? This RNA going into the processing, splicing and processing happens, right? So RNA which is formed, the part of RNA which is intron, which is non-functional, it is also formed through the DNA, right? But it is not a gene. That is here. Split genes. The coding sequence, one observes that eukaryotes, they have split gene, but the gene which is responsible for the formation of histone, it is non-splitting, right? Okay. Or expressed sequence are defined as exon. Exon are said to be those sequences that appear in mature or processed RNA. These are the functional unit. This one, exon, which can express. They are the exons are interrupted by introns. Introns are intervening sequence. Introns are intervening sequence. They do not appear in mature RNA or in processed RNA because through splicing they are removed. But only in case of eukaryotic RNA, right? The split gene arrangement further complicates the definition of gene in terms of DNA segment. That's why we cannot define a gene in the term of DNA segment. You understand? Because intron is not considered as gene. Inheritance of the character is also affected by promoter and regulatory genes. That is regulatory sequence of a structural gene. Promoter and regulatory sequence. We will read this one. Promoter and regulator means promoter, it is responsible for the attainment of RNA polymerase. Regulator means it is responsible for stop or start. We can say on or off, switch on or switch off the translation process, right? Hence, sometimes the regulatory sequence also are loosely defined as regulatory genes. Even though these sequences do not code for any RNA or protein, right? But they are also defined as regulatory genes. That's why what is genes? So when we use DNA, it is not very correct, right? So you understand we have two types of we can say genes are there. That is also we can say messenger RNA are there. Which one is? One is called monocystonic. So monocystonic means only one functional unit, right? That is present in only eukaryotes. Except which one? Histone protein. Right? Genes which is responsible for formation of histone protein, except this one. Second is our polysystonic. Poly means what? Many. Cystone means functional. It contains many, MANY, many functional units. Means more than one type of proteins are formed here, right? And it is present in prokaryotes, which is given here. This monocystonic, before processing, it has two things: intron, exon. Non-functional unit is called intron, functional unit is called cystone. Then in this case of splicing, we will read this one, right? When we read the next point, that is transcription, the process of transcription, right? But before going to this one, it is given types of RNA and the process of transcription. Okay, we will read this one. Now in bacteria, what is given? How many types of RNA are there? When we consider the type of RNA, we have three types of RNA. Look here, first we will describe the different type of RNA. When we consider the function of RNA, we have two types of RNA, that is genetic and non-genetic. First is genomic RNA, and this genomic RNA, it is responsible for inheritance, right? It is present in reovirus or TMB virus, tobacco mosaic virus, right? Sometimes it is called as virus, bacteriophage virus. Right. Second is our genomic first. Second one is non-genomic. Here we read non-genomic. Non-genomic means what? It is our transfer RNA. First, second one is our ribosomal RNA, and third is our messenger RNA. Now here we are going to read these RNAs. Non-genomic RNA, which is three types: transfer RNA, ribosomal RNA, and that is messenger RNA, which is responsible for the metabolic activity. So first here we have messenger RNA. Messenger RNA least means least ratio, least evident. Only 1 to 5% of total RNA, right? It is only 1 to 5% of total RNA. That means you say here, because they are most important, they have code. Why? Because after their function, after the complete their function, or after the translation, or after the formation of protein, they dissolve. They are only 1 to 5% of total RNA. Right. Now we have two types of messenger RNA. We can say One is our polycistronic messenger RNA and another one is our monocistronic messenger RNA. This one is present in prokaryotes and this one is present in eukaryotes, right? What is the difference between these two? First, when we see the structure, no G cap, no poly A tail is present. This one is polycistronic, but monocistronic means what? This side it contains G cap and that side it contains poly A tail. This is the processed RNA and this is G cap. We can say here 7 mg cap sometime also, right? This one is our monocistronic and this one is polycistronic, right? Okay. Now messenger RNA, who gave this name? Who gave the name of messenger RNA? It is the Jacob and Monod. Who give the name of messenger RNA? And Monod give the name, that is which name? Messenger RNA name. Right? Now second, these messenger RNA, they have codes, right? They have code for translation. And they are formed in nucleus with the help of DNA, right? Now second one is our ribosomal RNA. Ribosomal RNA. Ribosomal RNA, it is the most abundant. Most abundant means 80% of total RNA. It is. It is a structural RNA, means it is responsible for the formation of, it forms ribosome, right, with protein. Okay, so this is the structure, we can say structural tRNA. It provide platform for translation, right? It provide platform, ribosomal RNA. Okay, now how many types of ribosomal RNA? We cannot say types of ribosomal RNA. We can say, but how many types of RNA? Types of RNA involved in formation of ribosome. Because we have two types of ribosomes. One is called ATS and another one is called 7TS. ATS and 7TS, right? In ATS and 7TS, what happens here? ATS means it has two subunits. One is called 50S and the other one is called 30S. In 70S, it has two units. One is 50S and the other is 30S. In case of ATS, which is present in eukaryotic cell, one is 60S and another one is 40S. Here, two types of ribosomes, 70S ribosome first and another one is our 80S ribosome. 70S ribosome, it has two subunits. One large subunit is made up of 50S and the small subunit is made up of 30S. It has also two subunits, right? one is here 60s and another one is 40s. Now you can see this 50s, it is made up of two different type of RNA. One is here 5s RNA and another one is here 23s ribosomal RNA and another one is 5s ribosomal RNA. Right? 30s, it is made up of 16s ribosomal RNA. So three different type of RNA, they are responsible for the formation of a ribosomes, right? And here 60s, when we consider this 60s, it is made up of three different type of RNA. One is here, it is 28s ribosomal RNA, right? And 5.8s ribosomal RNA, and it is 5s ribosomal RNA. Three types, 60s, 40s, it is made up of 18s ribosomal RNA. Actually, these 23, 5, 16, 28, 5, 5.8, 5s and 18, when we consider total, one thing is response, this 5s is present in prokaryotic and 5s is present in eukaryotic. This one is common in both, right? When we consider in prokaryotic ribosomes, here three types, 23, 5 and 16s ribosomal RNA is involved in the formation of ribosomes, right? But in case of 80s, we have which one? How many? 4 types are there. 2.8 sRNA, 5.8 sRNA, 5 sRNA and 18 sRNA is responsible for the formation of a 80s ribosomes. So these are the ribosomal RNA which are responsible for the formation of ribosomes, right? And this RNA, ribosomal RNA, it was discovered by Kuntz, Z-E, Kuntze. He discovered this ribosomal RNA. Now, third one is our transfer RNA, which is very important also. Transfer RNA. Transfer RNA, two-dimensional. When we see this, if we see two-dimensional structure, then it is clove-leaf model. Right? But when we consider the three-dimensional structure, then it is inverted L-shaped structure. Is inverted L-shaped structure. Both structures are given by different scientists, right? As for two-dimensional structure, it is given by Holly. Cloverleaf model, it is given by Holly. But this three-dimensional structure, it is given by Kim, right? Actually, this three-dimensional structure is present in the we can say cytoplasm. Okay. To understand the structure of transfer RNA, we use this cloverleaf model, right? It has 4 arms. It has 4 arms, right? This type. In between these bonds are there, hydrogen bonds are there, right? One side it is 7 arms. 1, 2, 3, 4, 5, 6, 7 bonds are there. And that means 8 to 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Means 8 to 12. bonds are there. This side also 7 bonds are there. 1, 2, 3, 4, 5, 6, 7 bonds are formed. And this last point, it contains adenine, cytosine, cytosine. This side it is G, guanosine. Guanolic acid is there. Okay, we write here guanosine. We need not to write this one, right? Which one is this? This is the 5' end and this one is the 3' end. 3' means long arm is 3' end. And amino acceptor, this arm is called amino acid acceptor arm, right? Or sometimes we set them AAR, amino acid acceptor arm, because amino acid is attached with this A, amino acid attached with its COOH group, right? Group to 3' strand. And in 3' strand we know here which one is there? Adenine. Adenines, we can say here NH2. So adenines NH2 group is binds with amino acid COOH group. That means what? Amino acid is bind at that place. Do you understand? This side, no. This is the just opposite to amino acid acceptor arm. This is called anticodon arm. Anticodon arm. Due to the presence of inosine, this one is abnormal nucleotide. And due to the presence of this abnormal nucleotides, here means hydrogen bond is not formed. So a loop is formed. Formation of loop, it is due to the presence of abnormal nucleotides. Okay, here inosine is there, and these three codes, these are called anticodes. Anticode, right? So this, according to this anticode, we have 20 different type of transfer RNA for 20 different type of amino acids, right? According to these codes. Okay, these are anticodes, and these are responsible for recognize or identify the messenger RNAs. Now, this one is variable arm, which is variable. It is also called as variable arm because it is changed. It's changed, right? So because it's changed its structure, so it is called as variable arm. Now, one is C5, that is P psi 5C arm, P psi T psi C arm. Another one is called DHU arm or dihydrouridine arm, right? So here this one, because it contains 1, 2, 3, 4, 5, 6, 7, it is called as T psi C arm, right? Now you can understand this psi, it is responsible for pseudo uracil, right? So pseudo uracil, it is this one is like inosine. This one is abnormal first, this one is the second abnormal nucleotide. Due to the presence of pseudo uracil, here the loop is formed. And this is the DHUR, this one 8 to 12. This side 7 base pair, right? It is also 7 base pair. This side it is 8 to 12 base pairs are, and this is called DHUR, right? So DHUR, what is the full form of DHUR? DHU means what? Dihydrouridine. That is the third abnormal nucleotides. So due to the presence of these abnormal nucleotides, these are abnormal nucleotide. Due to the presence of these abnormal nucleotides, a loop is formed. Do you understand this one? Now, question is, what is the function of these arms? These arms, they have specific functions, right? As for example, T, that is our T first one is our T psi C arm, right? This one is responsive for, this is responsive for connection between, means it connect with ribosome. What is the function of this one is, it is responsible for the connection with ribosomes larger cell, right? Second DHU, this one is also called as amino acyl recognition site. Amino acyl recognition arm, because it recognizes the amino acid and also it is responsible for the attachment of amino acid, right? This DHU arm, it is an enzymatic arm. So this DHU arm is also called as enzymatic arms, right? So these are the three types of RNA, messenger RNA. We read here, you can see transfer RNA, then ribosomal RNA. I write here how many types of ribosomal RNA, eukaryotic and prokaryotic. You have to read this one. And then after this one is types of RNA, different types of RNA, means genomic or non-genomic. In non-genomic, we have to read here only non-genomic. Messenger RNA is also there. Okay. Now here the process of transcription. This process of transcription we will read in the next lecture. Okay. Thanks.